Okay, so let's talk about the cell cycle here and how the cell actually regulates and decides if it wants to divide or not. So the cell will actually spend the majority of its time in interphase, which is comprised of a few different stages, so G1, S, and G2, and there's also G0, which is considered more of like a stasis or dormant phase. And this all occurs before it enters mitosis. And there's also 3 checkpoints. So what does it mean when a cell is in G1? So let's draw a cell here. Actually, already have that. I'm just going to move it closer so you can see. So we have our cell, and when a cell is going into G1, that means that it's going to start growing. So G1 or growth 1. Okay. So let's make our cell bigger. Okay. So the primary objective of G1 is cell growth. Okay. At this G1 checkpoint here, the cell is actually going to check for DNA damage, because if the DNA has been damaged, it does not want to replicate damaged DNA and essentially copy all of those errors. So that's a big part of the G1 checkpoint. Another part is checking for cell size. Well, has the cell grown enough to have a large enough size in that which we are going to divide into 2 cells? There's going to be enough cytoplasm, enough nutrients, etc. for 2 cells, essentially. Okay, so that's what happens in the G2 phase, or G1. So let's just write that out. Put that over here. So the G1 checkpoint. So we're going to check for cell size and also DNA damage. And the cell also needs to be checking if it is appropriate to divide. Is it receiving cell division, positive cell division signals from its environment as well? Okay, so following this G1 stage, it's going to enter S phase. So once it passes this G1 checkpoint, the cell is now irreversibly committed to dividing. Okay, so this G1 checkpoint is really important. So if the cell decides that it does not want to divide, or that it is not an appropriate time to divide due to DNA damage, it doesn't have enough nutrients, or it's just not receiving enough signals, or there's not enough growth factors, etc., it'll enter G0. And that's kind of like a dormant, or like kind of like a stasis, like sort of like pausing that G1 stage there. Okay. But presume we did pass that G1 checkpoint, then the cell is irreversibly committed to cellular division. Okay, and we're going to enter S phase. So S phase, S stands for synthesis. So this is when the DNA is going to be copied. Okay. So we're going to go into S phase. And we're copying all of this DNA here. So I'm going to draw extra DNA in here. And notice that this DNA is actually still loose. Typically, when we're looking at, say, mitosis diagrams or meiosis diagrams, you see chromosomes like this in their condensed forms. So the DNA doesn't start to condense in interphase until we're actually actively in mitosis, okay? So the DNA here is still loose. Okay. Okay, so we finished our S phase. We've replicated all of our DNA here. Now we're going to enter G2. So G2 for growth. Okay. Drop this here. G2. And this is when the cell is just going to grow even larger. Okay. Remember, we need the cell to, at the end of it, be double its approximate size that it had started with. Okay, it's going to get even bigger. And we have also produced more organelles. Okay, so things like the centrosome, for example, would be doubled as well. So at this point, at G2, presuming we pass that checkpoint, that is when we're ready to enter mitosis. So let's go look at the next checkpoint here. So the next checkpoint, so we just finished G2. We're going to pass through the G2 checkpoint here. The purpose of the G2 checkpoint is mainly to check for DNA integrity. Okay? DNA integrity. It's so important that we're not making errors in our DNA replication. So that's why we have so many of these checkpoints, and it's honestly kind of redundant. Like the G1 checkpoint and the G2 checkpoint, we're both checking for DNA integrity and DNA damage. But the purpose of the G2 checkpoint specifically is that we're checking for faithful DNA replication. Because remember, we have passed that S phase. Okay. So I'm not going to go through all of mitosis, but I do want to indicate what that M checkpoint is for. So there's an M checkpoint right here. My mitosis video will be separate from this, because otherwise this video would just be way too long. Okay, so the M checkpoint is the last checkpoint. And when a cell is going through mitosis, it'll have condensed chromosomes, okay? So let's just draw it here. Okay, so suppose that is what our cell looks like at metaphase. So I'm just going to have the centrosomes here. We got the spindles coming out. Okay. So at this checkpoint, what we're doing is the cell is checking for that bipolar tension, okay? We want to ensure that the chromosomes are aligned properly prior to anaphase, before the chromosomes get split apart. So what the cell actually does is it actually checks for this bipolar tension, and it makes sure that it is even before it gives a signal to initiate anaphase, okay? So after that, then you have anaphase, and then telophase and cytokinesis, and you end up with two new daughter cells that are identical. So I'll go through mitosis in a separate video. This was just about the cell cycle and how the cell regulates that, and also the checkpoints. Okay, hope you found this helpful. We'll see you in the next video.